So before we actually proceed with the different things that will happen inside the mitochondrion, let's now consider first here what should be the prerequisite before the process known as the second half or other parts of the cellular respiration, which is known as the citric acid cycle and the chemiosmosis. Pyruvate oxidation will actually happen in the boundary of the cytoplasm and the mitochondrion. Pyruvate oxidation, it will happen when carbon dioxide will be released. Then, during the release of carbon dioxide, the pyruvate will also release hydrogen atoms, and it will simply be attracted to NAD plus to create NADH. Then, a coenzyme that will simply be utilized by the mitochondria known as coenzyme A will simply be attached to the pyruvate, creating now a new substance known as acetyl-CoA or acetyl coenzyme A. So once again, these are actually the different things that is happening during pyruvate oxidation. The first one, the release of carbon dioxide. The second one, it is now the reduction of NAD plus to NADH, and the attachment of the coenzyme A creating acetyl-CoA that will now enter the mitochondrion that will be utilized on the Krebs cycle, or also known as the citric acid cycle. So please take note. In the case of pyruvate oxidation, 2 NADH were created, 2 carbon dioxide were released, no ATP were created nor utilized. For phase number 2, known as the citric acid cycle, or also known as the Krebs cycle, it actually takes place inside the mitochondrion, and it will start with the presence of an oxidized pyruvate known as the acetyl coenzyme A. And the Krebs cycle, as summary, will actually supply the third stage of the aerobic respiration with electrons. Since the last part, we are now actually going to utilize here the different products that were simply created, known as the NADH and the FADH2 that will be utilized and created here on the citric acid cycle. Please take note that in every one acetyl-CoA, we are actually going to release here 2 carbon dioxide, the release of 3 NADH, then the release of 1 adenosine triphosphate and 1 FADH2 or charged FAD, or also known as the flavine adenine dinucleotide. Now, in the case of the citric acid cycle, we're actually going to utilize here a total of 2 acetyl-CoA. Since we utilize 1 glucose molecule, it created 2 pyruvate, then the pyruvate oxidation created 2 acetyl-CoA. So, it will actually be what would simply happen on the citric acid cycle. Now, as an overview, these are the different reactions that are happening on the Krebs cycle. Specifically, there are 9 different types of enzymes that will simply be utilized, but we are going to discuss them one at a time, just like how we discuss the concept of glycolysis. Now, to simply start the Krebs cycle, remember that there is actually a 4-carbon compound that is present in the cycle that will simply be recreated, which is now known as the oxaloacetate. If you can still recall, on the Calvin cycle, we utilized here the presence of ribulose 1,5-bisphosphate or RUBP. Its counterpart on the Krebs cycle is known as the oxaloacetate. Oxaloacetate will now actually attach itself to acetyl-CoA with the enzyme known as the citrate synthetase. The enzyme citrate synthetase will attach acetyl-CoA with the oxaloacetate, and the coenzyme A will actually be released to create now the very first 6-carbon compound present on the Krebs cycle, known as the citrate or citric acid. Okay? Citric acid is the very first 6-carbon compound that would be evident on the Krebs cycle. It is just the combination of acetyl-CoA plus oxaloacetate, which is done by the citrate synthetase. Now, the second and the third step, it is just actually being accomplished by the enzyme known as the aconitase. The enzyme aconitase, it is actually responsible for the process known as isomeration, in which it will just rearrange the citrate to create now a new substance known as isocitrate. From the name itself, isocitrate, it is an example of an isomer of citrate. Aconitase, it is actually an example of an isomerization enzyme that is responsible now for releasing water molecule and reattaching water molecule to transform these different types of substances from one compound to their isomer. In this case, a citrate will undergo the process of release of water molecule and reattachment of water molecule with the enzyme known as aconitase to create now the material known as isocitrate, which is just an isomer of citric acid. Then, the isocitrate will now actually interact with the enzyme known as isocitrate dehydrogenase. From the name itself, the enzyme is responsible for the removal of hydrogen that is present on the isocitrate. While the hydrogen ion is being released, it will actually be accepted by the NAD+ to create NADH. And while that is happening, the enzyme isocitrate dehydrogenase will also release carbon dioxide. So isocitrate will release hydrogen ions that will simply be accepted by the NADH. At the same time, the release of carbon dioxide, creating a 5-carbon compound known as alpha-ketoglutarate. Okay, from isocitrate, it will now create a 5-carbon compound known as alpha-ketoglutarate. And that is being accomplished on step number 4, which is the enzyme known as the isocitrate dehydrogenase. Then, the next one. The second oxidation will actually happen when the alpha-ketoglutarate will interact with the enzyme alpha-ketoglutarate dehydrogenase. The alpha-ketoglutarate dehydrogenase will do the following things, which is technically a lot. The first one, it is now the second oxidation or the oxidation of that ketoglutarate to release hydrogen ions that will simply be accepted by the NAD+ to reduce NADH, and carbon dioxide will also be released. Then, the alpha-ketoglutarate dehydrogenase will also attach the coenzyme A to create now another compound known as succinyl-CoA or succinyl coenzyme A. Okay, so once again, in the case of the alpha-ketoglutarate dehydrogenase, it will accomplish the following things. The first one, the oxidation of alpha-ketoglutarate to release hydrogen ions, and the hydrogen will be accepted by the NADH. Then, carbon dioxide will be released, and coenzyme A will be attached, creating now a new substance known as succinyl coenzyme A. And the coenzyme A will now actually be attached on carbon number 4. Now for the next step, the succinyl coenzyme A will actually interact with the succinyl coenzyme A synthetase to have the reaction known as the substrate level phosphorylation. From the name itself, the substrate level phosphorylation will actually utilize here an intermediate of adenosine triphosphate known as guanosine triphosphate and guanosine diphosphate or GTP or GDP. Okay? Guanosine diphosphate and guanosine triphosphate, these are actually intermediates that are usually utilized by different types of reaction that do not need a lot of energy. They are just there to simply assist. That is the reason why they are intermediates. Okay? You can observe here that the guanosine diphosphate will attach a free phosphate to itself with the presence of the succinyl coenzyme A. While that is happening, GTP or guanosine triphosphate will be created and the coenzyme will be released. Then, the phosphate that is present on the GTP will interact with adenosine diphosphate to create now adenosine triphosphate. That is the reason why it is known as substrate level phosphorylation, because basically a lot of things are happening on that certain type of enzyme. Succinyl coenzyme synthetase it is responsible now for the creation of adenosine triphosphate with the presence of the intermediate known as the guanosine diphosphate and guanosine triphosphate. Okay, and coenzyme A will be released, creating now a new substance known as succinate. Succinate, which is a 4-carbon compound, will undergo another reaction with the presence of the enzyme known as succinate dehydrogenase. The enzyme succinate dehydrogenase, it will now be responsible for the oxidation of the succinate by the release of the hydrogen that is present on carbon number 2 and carbon number 3. As you can observe here, since the succinate will actually release 2 hydrogen, it will now be accepted by another compound known as the FAD or the flavine adenine dinucleotide. The flavine adenine dinucleotide can accept a maximum of 2 hydrogen ions. Then the hydrogen ions will be attached to the flavine adenine dinucleotide, creating FADH2 or flavine adenine dinucleotide, which is charged, and will now create a new substance known as a fumarate. Compare the structure of the fumarate with the succinate. On carbon number 2 and carbon number 3 of succinate, the hydrogen ions were simply released. And in the case of the fumarate, that is the reason why double bonds were created between carbon number 2 and carbon number 3. I won't go to specific details regarding this matter, but remember those different types of byproducts because they will be utilized on the next phase of cellular respiration. Now, we have the product known as the fumarate. Then the fumarate will now react with the enzyme known as fumarase that will now have an attachment of water molecule. That water molecule will be simply split to hydrogen ions and hydroxide. The hydroxide will be attached on carbon number 2 while the hydrogen ion will be attached on carbon number 3, creating now a new 4-carbon compound known as a malate. Malate will now interact lastly with malate dehydrogenase, in which it will now be oxidized lastly, and the oxidation will happen on carbon number 2. The hydrogen ion will be released on the malate, and it will simply be attached to NAD+ to create charged NAD, or also known as the nicotinamide adenine dinucleotide, which is charged. And it will now create the starting 4-carbon molecule of the Krebs cycle known as the oxaloacetate. In the case of the malate dehydrogenase interaction with the malate, it will now regenerate oxaloacetate, which will simply start the Krebs cycle once again. Then acetyl-CoA will enter, then citrate will be created, isocitrate, then alpha-ketoglutarate, then succinyl-CoA, then succinate, then fumarate, then malate, then oxaloacetate once again. That's the thing that is happening on one acetyl-CoA enzyme that will enter. But if you're going to have 2 pyruvates that were created during glycolysis, 2 acetyl-CoA that will simply enter Krebs cycle, we are going to multiply this process by 2, having this summary. In the case of the Krebs cycle, how many NADH were created? For every 1 acetyl-CoA, there are 3. But in every glucose molecule, there will be a total of 6, since times 2. Then, the FADH2 that will simply be created would also be equivalent to 2, 1 each acetyl-CoA. Then, carbon dioxide, we have 4. Then, ATP that will simply be created, it is only 2 every glucose molecule. You can actually observe with a lot of processes that happen, only 2 ATPs were generated, because majority of the ATP will actually be present on the last step known as the electron transport chain, in which they are now going to utilize the electrons that were simply captured during the whole process of glycolysis, pyruvate oxidation, and Krebs cycle, which are present on the nicotinamide adenine dinucleotide and flavine adenine dinucleotide, which are both charged.